Hello again, everybody. Welcome back. Today we're going to spend a little time talking about how we can have variation of traits, or how we have different versions of all the traits that make us who we are. And if you recall from last time, we talked about how our DNA contains specific segments called genes. And within these genes are the instructions that we use to make proteins, and then the actions of these proteins give us our physical traits. And these are the things that make us different from one another. So for example, within a person's DNA, they might have a set of genes that code for the production of proteins, and the actions of those enzymes and other proteins might lead to them having blue eyes. The DNA itself does not contain genes for blue eyes, but it has the instructions for proteins whose actions create that blue eye color. And so we know that genes are used for the inheritance of traits, but we need to go into a little bit more detail about how these genes actually produce those physical traits. And so to talk about that, we need to mention a very famous scientist by the name of Gregor Mendel. And Gregor Mendel studied genetics. He's actually referred to as the father of modern genetics. He studied pea plants in the middle 1800s in Austria. And one of the characteristics of pea plants he looked specifically at was the color of the flowers. And so he noticed some interesting facts. He noticed that there were, with the flowers, there were two colors that these plants could have. They could be either purple or they could be white. And he noticed something really interesting is that more often than not, when he bred together a plant that had purple flowers with a plant that had white, the offspring would have far more purple flowers than white flowers. And it started getting him to ask some questions. And so one of the things that he started thinking about was the fact that somehow these parent plants were influencing the physical appearance of their offspring. And so for the purple flowers, we referred to them as with the letter P for that flower color trait. And so for the white flowers, we used lowercase p. And we'll talk about the significance of those capital and lowercase letters. We have learned since Gregor Mendel did his studies that for every gene, we have two copies of that particular gene in our DNA. One we inherited from our mom, one we inherited from our dad. And what we're talking about there is our genotype, which is what's in your DNA. And so if we use this flower color as an example, there's three possible genotypes for flower color. You could have two capital P's, you could have two lowercase p's, or you could have one of each. And so each one of these letters refers to what we call an allele. And so if our gene is flower color, the alleles are purple and white. So alleles often describe the traits that we're actually interested in here. Going back to our previous example, if the gene was eye color, the alleles would be blue and green and brown, things like that. Eye color is a little bit more complex than that, but it's an easy example to use in this context. So what does it mean if we have two of the same letter or two different letters? If we have two different letters, that condition is what we call heterozygous. And if you remember, that root word hetero means different. So if we have two of the same letter, that individual is what we call homozygous. So here we have a homozygous individual because there's two capital P's. Here's homozygous because there's two lowercase p's. And here's heterozygous because there's one of each. Now, where the capital and lowercase letters come in is referring to these traits as either being dominant or recessive. Now, if you remember, I said Mendel noticed most of the plants had purple flowers. Well, the reason for that is the purple allele is dominant to the white allele. So what does that mean? Well, all you need, or all the pea plants needed, is one copy of that capital P. If whatever the other version of that gene is that they inherited doesn't matter. If there was one capital P, then those plants had purple flowers. As opposed to white flowered plants, they have to inherit two copies of that white allele because it's recessive. The capital P kind of dominates or overpowers that recessive allele. So what that really means here is that in this situation, this version, the big P, And this version, the big P, little p, both of those are going to be purple. Whereas the little p, that individual is going to be white. In either case, what we're referring to is that organism's phenotype. So genotype is what's in our DNA, tells us what alleles we have for a particular gene, whether we're heterozygous or homozygous for that particular gene. And then that's going to determine whether we have the dominant or the recessive phenotype. And so again, here you see there's two ways that you can get the dominant phenotype, but there's only one way you can get the recessive. So another thing that goes along with that is if you say an individual is heterozygous for a trait, that's all you need to tell us because we know there's one copy of each allele. If somebody's homozygous or an organism is homozygous for a trait, you need to follow that up with either homozygous dominant or homozygous recessive. So where do these different versions of these traits come from, or where do the different combinations of these traits come from? And the answer to that is there's two main sources. There's meiosis and there's mutations. So we're going to start talking about meiosis first. Now you may be saying to yourself, that word sounds an awful lot like mitosis. And they are very similar, but there's some key differences. And I want to kind of review the highlights of mitosis that, in case you don't remember from previous classes. So in mitosis, you start with one diploid cell. And that one diploid cell copies itself and becomes two identical diploid cells. So these new cells are carbon copies. They're genetically identical to each other. And mitosis is used for growth and repair, meaning as you get bigger, as you get taller, you need more cells in your body. Those are going to be produced through mitosis. As you get older, your cells get worn out and they don't do their job as well. They need to be replaced. That's done by mitosis. In all of these cases, we're talking about somatic cells. So those cells that make up most of our body, the bone cells, the skin cells, all of the parts of our body that you're most familiar with, they get repaired and replaced through mitosis. Meiosis, however, is a little bit different. It's a little bit more complex. And so before I go into this diagram, let's talk about the overall process of meiosis. And so it starts very similar to mitosis in that we start with one diploid cell. But that's pretty much where the similarities end. Instead of ending with two diploid cells, we end up with four unique haploid cells. And by unique, I mean they all have different genetics or different DNA found within them. And meiosis is used to produce gametes. That is the only thing that meiosis is used for, is to create gamete cells. So we're going to produce the cells that are used for reproduction, and we're going to increase genetic diversity. So that's the part that I really want to focus on, is how we increase that genetic diversity. So let's take a look at this picture here. So first thing I want you to notice, right here, this is chromosome number 1. This is also chromosome number 1. The reason why they're different colors, we'll say this red one came from mom, and we'll say the blue one came from dad. And now in this cell, we also have chromosome number 2 from mom, chromosome number 2 from dad. And you may notice that they look a little different between this first cell and the second. What happened between here and here, the DNA copied itself. So I'll just use this as an example. This chromosome copied itself, and so each half of this X shape is identical to the other half. And so now this one became this cell right here, or that chromosome right here. So each one of these 4 chromosomes copied themselves. In the next step between here and here, you'll notice now we've gone from 4 chromosomes to 2. And the other thing that you'll notice is now this one is not all red and this one's not all blue. This one's red with a little bit of blue and this one's blue with a little bit of red. What happened is the 2 copies of chromosome number 1, so these 2 guys here, and then the 2 copies of chromosome number 2, these guys right here, they lined up kind of side by side. Oops, They lined up side by side, kind of like that. And they go through a process that we refer to as crossing over. So crossing over, or sometimes it's referred to as gene shuffling. So gene shuffling and crossing over are basically the same thing. And what's happening is, again, here's these two chromosomes. They line up side by side, and so parts of those chromosomes overlap with each other, and they swap places. So this little chunk right here swapped out with this piece right here, and this little piece right here swapped out with that one right there. So essentially what you're doing is you're taking chromosomes of DNA from your mom, chromosomes of DNA from your dad, you're shuffling them together, and you're dealing them back out just like a giant deck of cards. And the way this works is, in humans, we have 23 pairs of chromosomes. This example is only showing 2 pairs right here. Humans have 23 pairs. So we have 23 sets of chromosomes lining up side by side and exchanging genetic information with each other and then distributing that out. Considering the fact that humans have 30,000 genes approximately in our DNA, that means there are 60,000 alleles in our DNA. So essentially this whole process with our DNA is like taking 2 decks of cards that each have 30,000 cards in them, shuffling them together into one big stack of 60,000, and then dealing them back out into 2 stacks of 30,000. As you can imagine, the chances of getting 2 identical stacks in successive shuffling events is impossible. No matter how many times you take 2 decks of 30,000 cards and shuffle them together and redeal them, you're never going to get 2 identical stacks. And what that tells us is, no matter how many times this happens in our bodies, we're never going to create 2 gamete cells that have the exact same combination of traits. And so what this might look like in the real world, let's use some simple examples here. I'll say this is a chromosome. Whoa, that's a bad one. Let me try that again. Here's a chromosome from my mom, right? And I'm just going to make this up. This is all totally not true. But let's say that on this part of the chromosome, she passes along the gene to me for red hair, because my mom is a redhead. And then let's say she passed along the gene for green eyes. And then we'll add one more. There's a really common trait that people look at for genetic research, which is hitchhiker's thumb. If you notice, my thumb kind of bends backwards there. If some people, their thumb only goes straight. So if your thumb bends backwards like mine, you have hitchhiker's thumb. And then let's say that same chromosome from my dad. We'll just draw another one right here. Let's say he gave me the gene for brown hair. And then he gave me the gene for blue eyes. And then he gave me the gene for no hitchhiker's thumb. Now, when my body goes through meiosis and produces sperm cells, it's going to take these 2 chromosomes, it's going to shuffle them up together, and what might come out of it is a sperm cell that has the gene for red hair, blue eyes, and hitchhiker's thumb. So now I have a combination of traits that's being passed on to my kids and that combination was not present in either one of my parents. So I'm not creating new traits, but I'm creating new combinations of traits. And that's really important. It's not necessarily that this always produces new traits. Meiosis and gene shuffling produces new combinations of traits. Now, where we get new versions of traits comes from mutations. And so you may recall that a mutation is any change in the DNA. So for example, here we have a normal strand of DNA. When the DNA copies itself, it just splits down the middle. Each half acts as a template, so you can build a new strand. Sometimes in this process, mistakes are made. So here we have a T where there should have been a G. If that mistake doesn't get fixed, when the next round of replication occurs, this damaged copy is going to carry on that mutation. And every successive mitosis after that is going to repeat that mistake. That's the most common way that mutations occur. They can also occur during the production of proteins. So as we talked about previously, proteins are made in 2 steps, where the DNA is copied into a temporary piece called mRNA, and then that mRNA is used to go out into the cytoplasm and make a protein. In this process, sometimes mistakes are made and an improper sequence of amino acids is put together, or something happens in the copying process and the information gets jumbled up a little bit. In either one of those cases, there's 3 possible outcomes of those mutations. The first is that you might get some beneficial trait. You might get a new protein. You might get a protein that works better than it did before. It could lead to new physical phenotypes like camouflage. In this picture, there's one moth right here, and there's another one right here. Same species of moth, new versions of the trait caused by mutations. In the example of the cheetah, here we've got all sorts of mutations that ultimately gave them the abilities that they had. Cheetahs have a spine that's very flexible. They have claws that allow them to grasp the ground. They have long tails. All of these exaggerated traits that allow them to be successful hunters are products of mutation. And then same thing, sometimes these mutations will lead to new physical features for helping to attract a mate. So there's 3 benefits just in these examples here. In the first one, avoiding predators. In the second one, catching prey. And in the third one, attracting mates. Now, some mutations can also be harmful. You can create new traits or new color combinations that are not beneficial in the wild. The reason why you don't see many albino animals in the wild is it makes them hard to hide from predators or hide from the prey that they're trying to eat. Some mutations can cause drastic effects like extra limbs and things like that. And then other mutations can cause cancer and other genetic disorders. So the mutations in any situation, always keep in mind that you can have a benefit, you can have a harm, or you can have no change at all, what we call a silent mutation. And this is where, like in this example, the DNA has physically changed. Instead of CAA, our DNA is CAT, but the amino acid that codes for has remained unchanged. So this is what we call a silent mutation. The vast majority of mutations in our DNA are silent mutations. And so our next video will go into how we track these traits and how we predict the outcomes of different crossings in genetics. But for now, we're just going to leave it at that. So I hope you found that useful.